So along the x-axis here is time, and along the y-axis is the membrane potential measured in millivolts. The resting potential is number 1, and that is the membrane potential of a resting neuron. For number 2, that is the threshold, which is negative 55 millivolts for most living organisms, and they need to reach this threshold in order to undergo depolarization. Number 3 is depolarization, which is a reduction of membrane potential, which leads to an increase in membrane charge. Number 4 is repolarization, which happens when membrane charge decreases. Number 5 is hyperpolarization, also known as undershooting, and this returns the membrane potential back to resting potential. And then number 1 is back to the resting potential.
Okay, so let's take a closer look at what happens when an action potential actually passes through a neuron. In the structure of a neuron, you can see that we have a plasma membrane, and within the membrane are embedded certain transport channels that can either transport sodium or potassium. And the exterior of the membrane, or the extracellular fluid, starts off as more positive compared to the more negative cytosol. And this gradient is maintained by ungated channels not shown in the diagram and is used to conduct an action potential later on in this nerve impulse. And these gates are held shut by a mechanism known as an inactivation loop, which prevents the passing through of ions through these channels. And to start off, there's generally a much larger concentration of sodium ions in the extracellular fluid compared to potassium in the intercellular fluid, enforcing the positive to negative gradient. Now, in the second phase, called depolarization, some stimulus may cause some sodium channels to open, leading to some sodium ions entering the intercellular fluid. And if this depolarization reaches a threshold, an action potential may be reached. Now, if a certain threshold is reached, an action potential will be able to take place, and it'll enter the rising phase of an action potential. In this, most sodium channels open, causing a large influx of sodium into the cytosol of the cell. This makes the interior of the cell more positive with respect to its exterior. And if you notice, only the sodium channels open. The potassium channel remains shut. Immediately following this rise of action potential, the following action of action potential takes place. And in this, the inactivation loops block the Na+ inflow, and the potassium channels open, allowing potassium to flow out and keeping sodium on the exterior. This reverts the membrane potential back to its original form, in which its exterior was positive and the interior was negative. Now, in the final stage of undershoot or hyperpolarization, the sodium channel is completely shut, blocking the inflow of sodium, and the potassium channel is completely shut, blocking the outflow of potassium, returning the membrane potential to its original state found in stage 1.
Okay, let's take a look at a neuron. So the big sphere there is the cell body where most organelles and the nucleus are contained. It is followed by a long line called an axon where the signal is transmitted. At the end of an axon is the synaptic terminals where the signal is transferred to another cell. The branches around the cell body are called dendrites, where it is how the neurons receive signal from other cells.
Okay, let's take a look at a chemical synapse. So the circles that we're drawing are the membrane-bounding compartment where neurotransmitters are contained. They're called synaptic vesicles. Neurotransmitters are chemical messengers released into the synapse by the original neuron. Neurotransmitters can be both inhibitory and excitatory. Inhibitory neurotransmitters move the membrane potential away from the threshold, making action potential harder to reach, while the excitatory does the opposite. Between the two synapses is a narrow gap called synaptic cleft. It separates the presynaptic neuron from the postsynaptic cell. The presynaptic membrane is the membrane of the signal-sending neuron, while the postsynaptic membrane is the membrane of the signal-receiving neuron.